Good afternoon. I'm Matt Cartmill, and I want to welcome you to the fifth iteration of the Boston University Dialogues in Biological Anthropology. Today's session is entitled, Does Size Matter? Sexual Dimorphism and Human Evolution. Everybody has a practical interest in sex, but most of us are not very interested in the theory. But the theory is particularly important to evolutionary biologists because reproduction, the number of copies of a genome that get passed on to succeeding generations, is the only currency that counts in neo-Darwinian evolutionary theory. And most of this reproduction is done without sex because most organisms are bacteria, and bacteria reproduce asexually by fission. They have one copy of the genome per bacterial cell, and they simply make another one, zip down the middle, and wind up with two identical copies. So why should there be, in larger animals like ourselves, two sexes? And why should they differ in the ways that they do? We have two copies of our genome per cell. And like other larger organisms, we reproduce by producing a sort of giant specialized bacteria called gametes, cells that have only one copy of the genome inside them, and which fuse together to form a new individual with two complete copies of the genome. And this is sexual reproduction. We're all familiar with the basic arrangement of how it works. What makes it especially interesting from a theoretical standpoint is that organisms that do it usually produce two very different kinds of gametes. Great big gametes, like this one, are produced by half of the population usually, and those are the females. That's how you tell which are the females. They're the ones with the big gametes. The other half of the population produces little gametes, microscopic gametes. The big gametes are expensive to make, and the females therefore can't produce very many of them because a lot of nutrients go into each one of these, a lot of parental investment for the nourishing of the developing embryo and fetus. Males, on the other hand, produce very large numbers of cheap gametes, which contribute essentially nothing except the genome to the new individual. So the two sexes that result from this, males and females, have somewhat different interests and strategies in playing the age-long game of reproductive success. For females, the limiting factor on reproduction, the thing that limits the number of offspring they're able to have, is ordinarily food. Okay, the amount of nutrients that they can put into making copies of themselves, new individuals. For males, however, the limiting resource is the number of females that the males can get access to the favors of. And so this means that the two sexes have quite different sorts of interests and strategies. Females compete for food, males compete for females. When this competition takes the form of fighting between the males tend to get larger, and they may wind up becoming larger than the females. They may also develop special kinds of fighting organs or weapons like antlers or horns or the big canine fangs that we see in a lot of mammals, which are not seen or are smaller in size in the females of those species. And this is of particular relevance to human evolution because we belong to a group of animals, the catarrhines, the old world higher primates, in which sexual dimorphism is usually fairly pronounced. The males are always bigger than the females, and when there is a major difference between them, the males have bigger canine teeth and they're larger in body size. The big conspicuous exception here is human beings. There is modest dimorphism, that is two-shapedness, difference in average sexual dimensions, between males and females in body size, but essentially none at all in canine size, and what there is of no real functional significance. And so we're interested in how this came about, what it means, and what it tells us about the ways of life and the mating patterns and tactics of our remote ancestors. Here to talk to us about some of these things today are two first-rank specialists on the evolution and meaning of sexual dimorphism in primates. Professor J. Michael Plovkin from the Department of Anthropology at the University of Arkansas, and Professor Phil Reno from the Department of Anthropology at Pennsylvania State University. And they've come here today to discuss and debate some of these topics with each other and with the BU faculty, and at 4 o'clock with members of the audience here in the room. So I'd like to proceed without any further delay to introduce to you Phil Reno from Pennsylvania State University. Phil?
Thank you very much, Matt, and thank you, Matt and Kay, for inviting me and organizing this dialogues, and for Mike for being a co-participant in this. I've already had quite a number of great conversations today that have been very enlightening and fulfilling. So today I'm going to talk about a method we previously used to assess sexual size dimorphism within Australopithecines, specifically Australopithecus afarensis. And then I'll talk a little bit about how to put that range of size dimorphism that we see in that species into an overall interpretation of human or hominid evolutionary history. So sexual dimorphism in Australopithecus, particularly Australopithecus afarensis, has gotten a lot of attention, partly because of this kind of paradox of dimorphic patterns that we see from this kind of double, or this weird signal that we see within early hominids, that there's this reduced canine dimorphism. And here we see an Ardipithecus ramidus in a male canine tooth, which is actually substantially smaller than that what you see in the male canine tooth of chimpanzees. And this lower dimorphism, and in fact this feminization of the male canine, would suggest that there's been low intermale competition within the species, particularly for access to mates. In contrast, the body size dimorphism is still an open question. Many prior estimates have suggested there's been very high size dimorphism, and this has been led to conclude that there's been high intermale competition. However, because body size dimorphism, or estimating body size dimorphism, is actually measuring a characteristic of the population. And in fact, you're measuring characteristics of two populations, the size variation within males and the size variation within females. And so it's important to have a substantial sample of which to estimate these parameters. And then therefore, if you are restricted to a small sample size, you're often likely to have increased degree of error in your estimates. For example, if we look at kind of the initial views of size variation in afarensis, these are two of the earlier fossils found for the species. AL288-1 here, or otherwise more famously known as Lucy, has a relatively small size with a relatively small femoral head, where you have another specimen, AL333-3, is a much larger specimen with a larger femoral head. Here we have the measurements of these femoral head diameters, and we can calculate a simple ratio of the large specimen over the compared to the smaller specimen and come up with a ratio of 1.43. Now, if we compare that, we see that that's slightly larger than the ratio between, say, the smallest and the largest of a human femoral head diameters we see from a substantial sample of humans, approximately 50 individuals, but significantly less than the ratio that we see within gorillas. And it has been argued that if you can just randomly pick two Australopithecines and already get a size range much greater than, or slightly greater than you see in humans, that would suggest very large degrees of size dimorphism. However, with larger sample sizes or more fossils that have been discovered, particularly with looking at femora, which size can be inferred, we see that in fact this AL333-3 and Lucy remain to be the largest and the smallest of 8 afarensis femora. And in fact, looking at our analysis that I'll just discuss, these continue to represent approximately the largest and smallest specimens, looking at a sample of at least 19 different individuals. So we may actually also be seeing kind of approximately the size range within Australopithecus afarensis, and which may closely mirror what we see as the size range within humans, and much less than we see in gorillas. So the question is how do we attain a larger sample to get a dimorphism estimate? Our method that we used is what we call the template method. And the reason will become apparent in a moment. The problem we face is that we have a large assemblage of postcranial fossils from different parts of the skeleton from which size can be inferred, but it's hard to determine the relative size in relation to the total body of different parts of the skeleton. Say how big is as part of your distal femora or your knee compared to your elbow. Luckily, with Lucy, we have a partial skeleton from which the size relationships between different parts of the skeleton can be inferred from an actual member of this species. And so what we would do is use these size ratios to correct and get the relative sizes of the different fossils. So our method for calculating this, say, using Lucy as a template, would be to calculate a ratio where we take, say, AL333-7, which is a distal tibia. We can calculate a ratio relative to Lucy's distal tibia, multiply that by Lucy's femoral head diameter, and then we have an estimated femoral head diameter for AL333-7. And we can repeat this for other postcranial fossils, say the AL333-42 proximal tibia relative to Lucy's proximal tibia, multiplied by Lucy's femoral head diameter, gets us an estimated femoral head diameter for 333-42. Now, what do we do with these estimated femoral head diameters? Here we can see these estimates arrayed from the small femoral head diameters to larger femoral head diameters. And within this, now we can assess the relative variation within these different samples. And our method, one method we did we used was calculating the coefficient of variation, simple of measuring the relative dispersion relative to the mean. And for each of these different samples, or the way these samples are divided up based on how they're fossilized, we can group these based on those that come from the single locality AL333, which is interesting because it is a locality that has a very narrow time depth, potentially a single depositional event, that avoids many of the effects of temporal and geographic variation, which could also affect size variation within a sample. And there are 26 different fossils that have been found or we've used in our analysis from 333. In addition, there are other fossils that come from other localities, which we classify the non-333 sample. And this is useful because each of these fossils come from a different locality, and therefore they represent a separate individual. So we have at least 15 different individuals represented in these fossils. And then we just have the combined Afar sample, we call it, because all these come from the Afar region of Ethiopia. And this is a sample that maximizes sample size, looking at 41 total fossils. Now, for each of these, we can calculate a coefficient of variation, say 11.2 for AL333, 12 for non-333, and 11.9 for the combined Afar. Having a coefficient of variation is interesting, but it doesn't tell us anything unless we have something to compare it to. And so what we've compared it to is samples of extant species of chimpanzees, humans, and gorillas. And these are the kind of perfect samples to compare to Australopithecines. Not only are these the three most closely related extant species to Australopithecines, but they also basically represent the range of size variation with size dimorphism within primates, with chimpanzees having very little skeletal dimorphism. Males and females are very similar in size, males only slightly larger. Gorillas are very dimorphic with respect to their skeletons, with males much larger than females, and humans having moderate levels of dimorphism. So what we want to do is actually treat these comparative samples in the same way that the fossilization process has treated the previous afarensis population. And so we can treat the skeletons within these samples, either chimpanzees, humans, or gorillas, as a comparative sample, say containing about 50 individuals. And from that sample, we will randomly select a template specimen. This can serve as Lucy to give us the size ratios that are typical of the species in question. We then create a sample with metrics replicating the anatomical composition of the fossil assemblage. We can then use the template specimen to estimate femoral head diameters for each of these selected specimens. We calculate the coefficient of variation for that sample of estimated femoral head diameters. And then we repeat 1,000 times, and that therefore gives us now a range of variation of dimorphism estimates that are typical of coming from species of known dimorphism. Now, there's another issue that has to be addressed, a sampling issue, is the number of individuals at AL333. This originally dubbed the first family site, was formed from a nearly contemporaneous fossilization event. But what's interesting about it is from the dentitions, we can tell that at least 9 individuals were represented or somehow fossilized within this sample. And so therefore, there's the real strong possibility and likely possibility that each of these post-cranial fossils do not represent separate individuals. And many times you have one individual leading multiple fossils within our assemblage. So to correct for this, or to correctly model this, for when we're simulating the AL333 depositional event, we have, say, the sample of chimpanzees, humans, or gorillas. And from those, we randomly sampled 9 individuals to be the source of our metrics. And so from those 9 individuals, we select the 26 metrics to replicate the fossils at AL333, thus guaranteeing that many individuals will contribute multiple elements to the sample. In contrast, those that come from the non-3-3 assemblage can just be sampled directly from the total sample of 50 individuals, therefore making it a low probability that each of these 15 metrics will come from the same individual. And then the combined AFR simulation will just group the two different methods together in one simulation. Now, we can depict our results, for instance, using frequency histograms. And so here we have, for chimpanzees, humans, and gorillas, frequency histograms showing the number of times, say, a particular coefficient of variation was generated from a particular sample of one of these 1,000 simulations. And we can see that chimpanzees typically have relatively low levels of variation, humans in the middle, and gorillas with much larger ranges of variation. And here we have, in this black line, the afarensis value from AL333. We can also depict these as exact counts from our simulations. And we can see out of the 1,000 chimpanzee simulations, 122 out of 1,000, or about 12%, fell above the afarensis value. Humans, pretty much simulations fell on either side of the afarensis value, and gorillas, about 5% fell below the afarensis value. If we look and simulate the non-333 assemblage with 15 fossils representing unique individuals, we can see that, again, about 13% of chimpanzees fell above the afarensis value. Again, there was relatively equal probabilities of human samples falling above or below the afarensis value, and less than 5% of gorillas falling below the afarensis value. And then when we look at the combined AFR sample, which maximizes sample sizes looking at these 41 total fossils, we see that chimpanzees, less than 5%, fell above the hominid, above the afarensis value, and less than 1% of gorillas falling below. So from these, we conclude that it's relatively easy to sample the amount of variation we see in the afarensis assemblage from populations with human level of dimorphism, but significantly less likely to find that either sampling from low levels of dimorphism, like chimpanzees, or from populations with very high levels of dimorphism, like gorillas. So what does this interpretation? Oh, no, sorry, I just wanted to make one other comment on, because this dialogues is supposed to compare some methods between of the different approaches used, a recent analysis by Gordon et al. Came to a very different conclusion, suggesting that Australopithecus afarensis actually had displayed very high levels of dimorphism, either similar or even more extreme than gorillas. And they actually had a very ingenious method that I was quite impressed with, which I don't have the time to go into the details right now, but using a property of the geometric mean, they were able to calculate separate dimorphism ratios for different anatomical locations and combine them within the same test statistics. But one key step of their analysis was to assign individual fossils a particular sex. In this case, in this plot, the blue ones were those assigned to be males, red ones assigned to be females, and they were assigned based on whether or not the fossil value fell above or below the average for that given metric. So for instance, for the humeral head, there are only two specimens. The smaller one was assigned to be female. The larger one was assigned to be male, and a ratio was calculated based on those. However, say for the femoral shaft, there were two that fell below the mean of these 6 specimens. They were assigned to be female, and these other 4 were assigned to be male. Now, what's interesting is if you compare that to our estimated femoral head diameters and see where these specimens fall within their relative size, we can see that the vast majority of the data that are contributing to female size estimates all come from 2 specimens, and those are Lucy, or another partial skeleton, AL128 and 129. And in fact, these are the 2 smallest specimens within our analysis. In fact, this other fossil that is marked blue is AL322, distal humerus, which we actually had a number of choice of 3 metrics to judge its relative size to Lucy. We actually ended up choosing the one that gave the smallest size estimate. In fact, the other metrics suggest that it actually is quite substantially bigger than Lucy. Probably more appropriately gives a estimated femoral head diameter of about 32 millimeters. And if we had chosen one of those metrics, it would actually substantially reduce the coefficient of variation for this assembly, for afarensis. So it actually is kind of conservative to leave that specimen here. So regardless, basically standing in for this analysis, we have basically 10 of the 11 data points for females are all coming from these 2 specimens, which are at the very low size range for afarensis. And these 2 specimens are standing in as kind of the typical sized females. And so if, in fact, maybe the female size range was substantially larger, maybe much lower levels of dimorphism would be attained for this sample. Now, what do we make with a conclusion or suggestion of moderate size dimorphism for early hominids and also for humans? Well, it's important not to get too focused on one human-specific trait. In fact, there are a number of key human and hominid traits that differ between us and other apes. We have much larger brains and a capacity for advanced behavior. We are bipedal, which is a very bizarre form of locomotion. We have a very number of dietary differences between us and apes, and particularly between our hominid ancestors. And we also have a unique suite of sexual characteristics that are specific to males and females and that are very different from the suites that you see within other apes. And so it looks that there actually might have been a particularly hominid reproductive suite during human evolution. We know from the fossil record that there was this kind of feminization of the male canine. We can also, from our results, suggest that there was moderate skeletal dimorphism. But also, modern humans have a number of soft tissue characteristics that are associated with our reproductive behaviors. Human females have, compared to other primates, a very bizarre pattern of having concealed ovulation, not only concealed from males, but actually concealed from themselves. Females have permanently large mammary glands, which might serve to actually conceal when they re-enter the reproductive cycle after lactation and after weaning an offspring. And there's also been actual dramatic olfactory receptor loss in humans beyond what you would just expect through drift that may have one possible evolutionary significance of that is actually choosing males that are actually more oblivious to female reproductive cues. In addition, males have experienced probably likely reduction in sperm competition. Relative to the other apes, we have moderate testis size, small seminal vesicles, and inability to produce vaginal plugs during copulation. So we're not engaging in sperm competition. Now, if you look back at the fossil record, we can see that the reduction of the male canine tooth is one of these cardinal features of the hominid lineage, evident even in Sahelanthropus at over 6 million years ago. By about 5 million years ago, Australopithecus suggests that we have begun to be a habitual biped with continued canine reduction. By the time we get to Australopithecus, it appears that we were, by 4 million years ago, an obligate biped with a very kind of modern style of bipedal locomotion and further reduction of the canine. And then by about over 2 million years ago, and even signs of it within Australopithecus, evident brain size expansion that continued up to modern humans. And so as many of these human-specific characters actually have a very ancient history within our hominid lineage, it might also be possible that some of these soft tissue reproductive characteristics also have an old history. Now, the one last feature that should be mentioned that is a unique hominid trait is our transition from a demographic dilemma that faced our ape relatives. If you look at this graph here, we can see in red all the localities where apes fossils were found in the Miocene, not only throughout Africa, but also in Europe and Asia. However, in modern times, apes are really only found in a few kind of basically tropical refugia in Africa and Far East Asia, while in contrast, monkeys have kind of replaced in many of the localities where apes had been previously found. And so from the Miocene to the present, we've had this reduction of ape species with this great expansion in the replacement with the number of monkey species. And this might result from apes having very low reproductive rates, typically with apes producing only one offspring at a time and only once every 5 to 7 years, whereas monkeys can produce one offspring but every 2 to 3 years. And so if they were to tackle the same niche, monkeys could actually just replace apes through their increased demographic success. In contrast, throughout the Pliocene, Australopithecines seem to be expanding. Australopithecus afarensis was found throughout East Africa and into Central Africa and Sub-Saharan Africa. And even by the time we get to later Australopithecines like Africanus, spreading into South Africa as well. And so the question is, how could our ancestors be expanding while our relatives as apes were reducing? One possible scenario would be a reconstruction of reproductive behaviors involving pair bonding between males and females, with males actually actively engaging in provisioning their offspring. This would accord with the evolution of bipedality, which would require extensive carrying of high-value, low-frequency food sources, particularly those valuable foods and fats and proteins that would be very beneficial to a female's nutrients you'd need during reproduction. We'd have canine reduction, which would result from female choice, picking males who are actually less competitive in the agonistic competition for polygynous mates. We'd have concealed ovulation selected by males who would prefer females who did not display their reproductive status, and so they could actually have better paternity assurance. Moderate size dimorphism could evolve as males and females engaged in different niches for foraging, particularly as males would have larger ranges to forage from places where females would not forage themselves. And then ultimately an increased reproductive rate, which would result in a reduced interbirth interval. You'd eventually, with two parents engaging in a reproduction, you could actually extend the juvenile period because you could now overlap dependent offsprings. And this extended juvenile period would allow for periods of extended learning and therefore allow selection to operate to increase brain size. So with that, I will conclude and just want to thank a number of people that have helped on this research along the way.
Thanks very much, Phil, for laying out that body of theory and that sort of comprehensive picture of what early human ancestors and their evolution may have looked like. For a somewhat different take on some of these topics, now I'd like to bring up to the podium Professor J. Michael Plovkin from the University of Arkansas.
Thanks, Matt, and thanks, Kay and Phil, for coming and talking about this stuff. As Phil mentioned earlier, we've been talking already today about this, and it's been very productive and quite a lot of fun. And let me emphasize on this that the most important aspect of what we're discussing here is the amount of fun that we have with it, because science is not so much fun because we can figure out a single problem, but rather because we can have these wonderful debates about problems, and it's the real problems that make the thing interesting. The simple adding 2 and 2 together to discover that you've made 4 is useful, but the real fun is getting at the intractable problem. And sexual dimorphism in human evolution counts, I think, as an intractable problem. People have known, or suspected, I should say, for a very long time that early hominins are sexually dimorphic in body size, and they know that primates are sexually dimorphic in canine tooth size, and Darwin had originally speculated on the reduction of the canine teeth in humans. And modern humans, of course, show a very modest degree of size dimorphism, and when fossils began coming out of the ground in East Africa, it became apparent that they might be very strongly size dimorphic, even though their canine teeth are not dimorphic. Now, the reason that people care about this is, first of all, taxonomy. If you have a lot of sexual dimorphism in the fossil record, it implies that there may be more than one species. But we'll leave that intense and furious debate aside and focus on the other option, which is that there are actually 2 sexes present in the fossil record. If sexual dimorphism is related to sexual selection, it is also related to social behavior, and people recognized very quickly that sexual dimorphism, or the presence of it, would have implications for social behavior. And as a consequence, although there are as many models for the evolution of human behavior as you can possibly imagine, sexual dimorphism in size and in canine tooth size constitutes some of the only actual evidence, direct morphological evidence, that may be associated with behavior in the fossil record. So it becomes an argument, a very contentious argument. Now, people have used size dimorphism in the fossil record to reconstruct monogamy, human-like mating systems, chimpanzee-like mating systems, bonobo-like, promiscuous, gorilla-like, multi-male, intense male competition, which covers pretty much the entire primate range. The first conclusion as an outsider to draw from this is that dimorphism is pretty much useless for inferring anything about behavior, which I will say as somebody who studies this is partly true. However, there are other ways around it, and we understand quite a bit more about sexual dimorphism and its evolution than we did in the past, and there may be ways around this very knotty problem. But the first part of the problem is getting at how to estimate the magnitude of dimorphism in the fossil record. How dimorphic were early hominids? As Phil just mentioned, he presented a model in which he reconstructed Australopithecus afarensis, which is one amongst a number of hominins, as being human-like in its dimorphism. Others have stated that the dimorphism is somewhere between that of a chimp and a gorilla, and others that it is gorilla-like dimorphism. Now, saying that they were of any magnitude of dimorphism is a loaded term because we're talking about different kinds of sexual dimorphism. The first is that for the fossils, we only deal with skeletal dimorphism. Cranial, dental, and skeletal. That's all that we have. But when we talk about sexual selection, we're talking about canine teeth and body mass, because males do not compete with one another by standing there at the bar and ripping open their femoral head and showing it to another male to impress him with how large it is. It's your body size and your body mass that count. And it's that relationship between body size and skeletal dimorphism that can cause all sorts of problems with these kinds of analyses. The first question to ask for this particular talk is, what exactly do we mean by human-like dimorphism? This is something that actually doesn't get addressed very much in the fossil record in the discussions, because it turns out that humans are rather odd in their pattern of body size dimorphism in canines. It's not just that we have some size dimorphism and some canine dimorphism, it's that we have a different relationship between our cranial, our skeletal, and our body size dimorphism. Now on this graph, what I've plotted here, there we go. What I've plotted here on this graph is a ratio of male to female body size. This is a log scale for those of you who are confused about what those numbers mean, so just ignore it. Just know that animals that are down here at the low end are not very dimorphic. Males are not much bigger than females. Animals that are up here are very dimorphic. And we have gibbons, which are not very dimorphic, chimpanzees, orangutans, and gorillas over here. Now let's plot out some patterns of human dimorphism to show what it looks like. If you just take a bunch of humans and put them on a scale and measure the ratio of the male to female size, the total mass dimorphism of humans comes out as between those of gibbons and chimpanzees. And it's not a lot. If you look at stature, and a lot of anthropologists use stature to measure sexual dimorphism, and you adjust that stature to make it equivalent to body size, and I'll discuss that in a moment, the stature dimorphism comes out as slightly greater than the body mass dimorphism. However, if you take the humans and you boil off all the body fat, and you just use what is called the lean mass dimorphism, you'll find out that now they fall out as somewhat greater than a chimpanzee. So human size dimorphism varies depending on whether you're looking at the total body mass less the body fat, or the stature, and we can go through some other characters here too and show that there's variation in these things. Now, another thing here is to not just realize that there's variation in the pattern of human body composition dimorphism, but also we need to place this within a broader context. Extant apes represent a very small number of species of the total number of primates, and our models for the evolution of dimorphism are based on primates. So let's place humans within the primate context. In these two graphs, I have body masses that have been derived from about 140 species of primates, and I've divided them up into two common categorical estimates of that are supposed to be related to sexual selection. The first one is monogamy and polygyny. These animals are not supposed to be very dimorphic because males do not compete much for females. We then have multi-male, multi-female groups, which are supposed to show intermediate levels of dimorphism. At this end, we have single male, multi-female groups where males supposedly exclude lots of females or other males from access to females. And then we have species with mixed social systems. And you'll find out that there's a lot of variation in dimorphism in all of these mating systems. The monogamous and polyandrous species tend to show small degrees of dimorphism. Over here we have competition levels, which is another way of estimating, an attempt to estimate the magnitude of male competition. Species over here on this end, males are not supposed to be very competitive, and on this end they're supposed to be very competitive. It's just another way of doing this, and we get a pattern where species that compete more on average are more dimorphic than those that don't. We also have here monogamous and polyandrous species tend to not be monogamous, whereas polygynous species tend to be all over the map. Now, where do humans fit on this? We'll place the body mass dimorphism in here, and you'll find out that they are consistent with every single competition level and every single mating system. It is low, but you can find a primate that's like that that competes in any way that you want. Stature dimorphism, same. What about the body mass, or the lean mass dimorphism? The lean mass dimorphism skirts at the upper edge of the monogamy or low male competition range, but still is about in the middle of the pack. One problem I'll note about using the lean mass dimorphism is that it is a little bit akin to saying that when we remove the parts that are not dimorphic, those parts that are dimorphic are remaining, and therefore it must be dimorphic. So there's a problem there we have with that kind of data. But this gives you a sense of the issue with human dimorphism. It is not uniquely associated with any particular behavioral pattern in primates, so you can't really say much about it. That's a problem. Now, turning to estimates of dimorphism in hominins, if we take a magnitude of body mass, skull, postcranial, and lean mass dimorphism in humans, what I've done here is listed out where I got the data from, but these are pretty typical values. If we look at body mass dimorphism and you adjust to make everything proportional, because we don't want to look for differences between linear and volumetric measurements, human body mass dimorphism is about 5% males bigger than females, adjusting for it's the cube root if you need to know it. The skull dimorphism is about 5%, in line with the body mass dimorphism. The postcranial dimorphism is about 14%, a ratio of males to females 1.14, which is in line with the lean mass dimorphism of 1.13. Now that's particularly interesting. To note on this, though, if you look at other primates and you look at the skull and the postcranial dimorphism, they will generally match. Humans are uniquely different, or nearly uniquely, in that the skulls show particularly low dimorphism and the skeletal particularly high dimorphism by comparison to each other. If we look within the Australopithecines, here are some estimates, various estimates of skull, mandibular depth, mandibular breadth, postcranial dimorphism, ratios of 1.1, 1.2, 1.16 for mandibular breadth. The postcranial dimorphisms are from, in this case, the original 2003 Reno et al. Estimates, and these are the new estimates that Phil just had presented. Instead of using CVs here, I'm using the other thing that they did, which was the reconstructed ratios, or 1.12. It's about the same. It stated that adding the new data in do not make a difference between the 1.16 and 1.12, which I would argue, when you first look at that, yes, that seems to not be particularly inflated dimorphism when you toss the new specimen estimates of these things in. Notice, however, that the skull and the postcrania match in their estimates. If you look at Africanus, skull dimorphism about 1.2, 1.12, depending on who it is that you use. The postcranial dimorphism, very few people have actually put a number on that. Elizabeth Harmon and Green did a thing where they basically said it was slightly more than a chimp. But in that same analysis, Green also noted that their analysis found in the same analysis, in the same paper, that Afarensis was considerably more dimorphic than the Africanus that they were looking at. For Boisei, since we have no postcrania, we just have skulls, it's about 1.15. So if you're looking for a magic number for hominids, let's imagine 1.15 for the moment and ignore the variation, and let's work with that number because it's in line with what Reno et al. Had presented with their template method from 2003 and say is stable. Here are all the estimates of sexual dimorphism of Afarensis from the template data that Reno et al. Used. And you'll notice they float all over the map here, but the magic ones here are the 333 only site, because this should show the least degree of temporal and geographic variation, confounding the estimates. What I've done here is used different methods to estimate dimorphism. There's lots of different methods, and they all give about the same results, so you don't have to worry about the details of it, but they're about the same, 1.16, 1.17. If we add in the new data to the 333 only site over here, the estimates are inflated to about 1.2 to 1.21. But what does this mean? Well, there's an implication here. These estimates are all linear estimates of dimorphism. That is, they're like measuring a skull length, and a skull length is just a linear estimate from here to there. It's a ratio of a line. If you want to make that estimate comparable to an area, say tooth size area, you have to square that value. So if, for example, in this animal that I've used, you have a skull dimorphism estimate of 1.15, you'll find that if everything is isometric and proportional in the animal, its tooth area would have 32% dimorphism, and its body mass would have 52% dimorphism. 1.15 is the skeletal dimorphism estimated in Afarensis, but that's equivalent not just to human-like dimorphism potentially, but also to guenon and macaques and baboons and nasalis, and lots of animals, in fact. So it could be strong, or it might not be strong. If you go back to the original data, and here at the bottom I've added to that chart estimates of body mass dimorphism, and all I've done here now is cube all those values, those linear values, to give a proportionality to body mass dimorphism. The value of 1.20 from the new template method that was published a year or two ago implies body mass dimorphism of about 73%, which, if you compare it to humans, is either like it or not like it. It is actually hard to say. Okay? The important thing is here that it is related to size dimorphism, and it's that relationship to size dimorphism that is ultimately going to determine its biological relevance. Now, really what's at issue here is what the best method is to estimate dimorphism when the sex is unknown, because the problem here is we don't know the sex of anything, so we have to guess it, and it's a problem. Now, the arguments that are here is, first of all, which element is best, skeleton or skulls? A number of people, not just Reno et al., but lots of people, say that the skeleton is better than the skull. I disagree right up front, because my data say that they're both about equivalent. I'll show you that in a moment. And the second is, which method is best? A multivariate method, which is the Reno et al. Method and the Gordon method, which Phil had also reviewed there, or a univariate method based on a single dimension. Now, first of all, which method is best? Which skeletal element? This is sort of a classic relationship here in these two graphs. Over here on the right side, I have plotted female body size against femoral head diameter. Femoral head diameter is usually acknowledged as the best estimate of size across species and even within species, and you will look at that graph and say, wow, it is tightly correlated with size. It is a good estimate of size. Over here we have jaw length. Jaw length is poorly correlated with female body size. Same species, but now we're using jaw length. Down at the bottom here I have the percent prediction error. That gives you an estimate of how accurate the estimate is. The lower that number, the better the estimate. So the percent prediction error for femoral head size is low. It would imply that it's a better indicator of body size. So if it's a better indicator of body size, it should be a better indicator of size dimorphism. Rarely, though, is that actually evaluated. So here I've done it, the same data, an estimated body size dimorphism against femoral head size dimorphism and jaw size dimorphism. And now the femoral head size dimorphism is actually a poorer predictor of body size dimorphism than the jaw length dimorphism, and that will hold whether you use the individual dimensions or the multivariate dimensions. This does not mean that skull dimorphism is a better estimate of size dimorphism. It simply means that you can't dismiss skull dimorphism as being irrelevant and that you should prefer one over the other. It means that both are providing information. Now, if you look at the methods, the template method versus other methods, what I've done here is a large analysis, basically repeating what Phil had done in his analysis, so I won't go through the details of it. But what I did this for was 105 species of primates. And I reconstructed the template method and a series of other methods, and then regressed the known dimorphism, the values of the known dimorphism, against the estimates of dimorphism that we've generated using the different methods in living species. I also did this for a number of univariate methods, that is, methods that evaluate a single dimension. In this case, I just looked at skull length as representative. The numbers to look at on this graph are here. This is the correlation between the known dimorphism and the estimated dimorphism. For the template values, they differ, depending on whether you use the mean or BDI method, between 0.71 and 0.79. The geometric mean method of Adam Gordon gives a correlation of 0.92. The individual measurements give you much higher correlations, which with my reading glasses on I can't quite see, but it's around 0.98 or so, very high. Another sense of this for the accuracy of the method is to look at the standard error of the estimates from these regressions. The standard errors are 0.036, 0.027 for the template method. The others give 0.012. What this is actually telling you, if you look at those numbers, is that the template method is 3 times worse at estimating dimorphism than the other methods. It has tremendous amount of error that's associated with it by comparison to the other methods. So how does this impact our impression of dimorphism? Well, what I did was I used some Paranthropus boisei skulls, and the reason I use skulls is because I got just enormous amounts of skull data, but it should give us exactly the same validity for evaluating the methods. And I used a comparative primate sample, and from this comparative primate sample in this graph, I've shown what the skull dimorphism looks like, the true skull dimorphism from the primates. And I divided it up with these competition levels just to sort the data out as a behavioral estimate. I then estimated Paranthropus boisei skull dimorphism using the template method and plotted it against those data that have now been calculated. You see the numbers have shifted on the axis, and these now are the primate data that have been calculated using the same method. So now we're comparing the fossil estimate to extant estimates based on the same methodology so that they're equivalent. Paranthropus boisei by this method has very low dimorphism by comparison to other primates. Let's use Adam Gordon's method. Gordon's method has a little bit more segregation, it seems, and not quite so much inflation. The estimate of dimorphism in boisei skull is higher, and you'll notice that it's now consistent with the high competition species and not just the low competition species. So these methods are giving us very, very different signals. Now, if you compare the data summary methods and just look at exactly what's going on, what are the data summary methods doing to the data? All I'll do here is just superimpose the estimates of dimorphism from our comparative sample to the known dimorphism from those same samples that were used to generate the estimates. There's the template method on one side on the left, the geometric mean method on the other side. In red here, these are the true estimates. And what you see is that the low dimorphism species have had their dimorphism inflated substantially. The same happens with the geometric mean method, but it doesn't happen quite as much, and that is reflected by those previous correlations that I showed you before. The method is slightly better than the template method, but I will emphasize that all of these methods inflate the dimorphism for low species. They all do it. They do not provide necessarily accurate methods unless the dimorphism is very, very strong in these animals. So this is a constant problem that we face in estimating dimorphism in fossils. So given all of that, which undermines my confidence certainly, then what do we say about models for evolution of dimorphism in primates? Well, the models for dimorphism in primates have come a long way since people have originally implied that they were monogamous if they didn't have any dimorphism and polygynous if they did. Dimorphism is now recognized as independent selective factors that work on males and females. Male body size and female body size are a product of a unique regime of selective factors favoring changes in size of one sex versus the other. There's nothing that selects for dimorphism. Males are relatively straightforward and simple. Dimorphism is a function of male reproductive skew. How much variation in reproductive success do males get as a result of fighting for one another? Male competition is what leads to the reproductive skew. And it's the monopolization potential of the females that leads to competition of males. Males will compete for females when they can, and when they can't, they won't do it because fighting is dangerous, and if it's not going to get you anything, the animals won't do it. And then impacted on that are things we call female counter strategies, because the females generally will try to resist strategies that are not beneficial to them. So there's a complex dance that males and females do here. Generally speaking, male body size is a factor of ecological size differences, and then on top of that, it's male competition. That is generally conceded that large male size is favored by sexual selection in association with competition for females. Female size, though, is more complicated. There is a phenomenon called correlated response to male traits. Since the genetics control these things, if you select for large male size and there's nothing to dissociate male and female size, since we all share the same genes, then if you make big males, you'll make big females. Now that factor is known to be, or thought to be, not a particularly strong factor, at least within primates and several other animals. But selection can favor larger females. Due to what? Very briefly, you can get big females because they compete for resources, which has been shown in some cases to be probably true. Lower female or infant mortality. Bigger mothers make better mothers because infant mortality and female mortality are lowered with larger females. This is also known to be true in some cases. Selection can favor smaller females. It can do it because smaller females breed faster and potentially can increase reproductive success by shooting out more babies. Earlier breeding. If you mature earlier, you get out of the immature phase quicker, lower your mortality, and can start reproducing earlier. Or you can lower your metabolic demand because big females require more food in very seasonal environments or food-limited environments. It can be advantageous to be smaller. Now, in reality, we don't know what affects female size. We know that all of these factors can affect it. But at this point in our knowledge, we just don't know because no comparative analysis has been able to tease these factors apart. We just know that they can be there. So where does this leave us for the fossils? How do you interpret dimorphism with this stuff in the fossil record? Well, I will be very speculative here. And let me emphasize here that I don't care how dimorphic any hominin is, and it's all interesting. But we can map these things onto a phylogeny and do a heuristic exercise, so to speak. Gorilla has strong sexual dimorphism. Pan has moderate size dimorphism. Australopithecus, we'll assume for the moment, has strong size dimorphism. Paranthropus, we'll assume, also has strong dimorphism. Ardipithecus appears to be slight to modest dimorphism at most, or monomorphic. And sexual dimorphism in Homo we'll call weak for the moment. What would this imply? In this phylogeny, if Ardipithecus is truly a hominin ancestor, and if it shows no dimorphism, which is perfectly reasonable, it would imply that Australopithecus became size dimorphic by a reduction in female size. It would imply that Paranthropus became size dimorphic by an increase in male size. And it would imply that Homo became modestly dimorphic through an increase in female size, without a concomitant increase in male size. Now, what does all that mean? Well, this turns out to be hard to interpret, of course, but let's imagine three scenarios here very quickly. Let's say that we have a shift in female size from small to large, or large to small, it doesn't really matter. In most primates, what we generally find is that when females shift size, the dimorphism remains proportional. In other words, males and females will change size in lockstep with one another. Females get bigger, the males get bigger. And this goes along with the general idea that if female body size is optimized to the environment, male primates tend to live, act, sleep, mate, eat the same things. They live in exactly the same niche as the females. And therefore, they suffer the same pressures, selective pressures on body size that the females do, but they also have another selective pressure for increased size in association with competition. But that increased size, if the female is optimized, represents the optimal size, means that the males will also face a cost. And so this sliding up and down of keeping dimorphism the same simply represents a balance of costs and benefits for changing male and female size relative to one another, as long as the sexual selection is the same on the males. Now imagine we could look over time and document changes in male size only. Say males get bigger, but the females don't, or that the females get smaller. All that you could say from this, all that you could say, is that there's a shift in sexual selection on male size. Nothing more, and you cannot infer the cause. Finally, let's take a look at a shift in female size associated with a shift in female monopolization potential. In other words, the female body size is changing, but what if the male size doesn't? This has often been portrayed as the dimorphism is lost strictly through a change in female size, but this model would say that no, The female size is changing, which is changing the dimorphism, but the failure of the male to keep in step with the female means that the females are changing something so that the male is also, sexual selection on male size is also changing. And so this lack of keeping pace with female size is also a dynamic process. And in this case, all you could say is that the females are changing their size, and in association with that, something is going on to change selection on male size. An example of this, it is commonly thought, although it is not actually known, and we can discuss this later, that Homo erectus lost size dimorphism through a shift in female size. Over time, females got larger, and males did not get as large. Well, what would this imply? It changed in selection on female size, but it also affects selection on male size. And so you could map this out as a shift in female resource acquisition, or something like that, would lead to an increase in female size. At the same time, it would be associated with a change in female group size and grouping patterns, which would lead to a decrease in the female monopolization potential by the males. They simply can't fight and get differential access to females, which would lead to a decrease in reproductive male skew, which in turn would result in a decrease in dimorphism. That's about it. But of course, the problem with that is we need a fossil record. And the reality is that for most of this stuff, we really don't. And that's where the issue comes in, is these debates are based on a very, very thin fossil record. And it leaves open a tremendous amount of speculation. So that'll end it. Thank you.
Thank you, Mike. We have about 5 minutes left in our webcast this afternoon. I would like to reiterate that we'd like to have you all come on over and help the rest of us ask penetrating questions of our two guests from out of town. We haven't got a lot of time left for discussion and questioning at the moment, but there are one or two questions that I would like to raise in connection with the interesting models and numbers and statistical distributions that we've seen in the two talks that went before us. And so let me just ask you two folks this. And I'm going to start by asking Phil. I'm going to sit down over here with him just a couple of minutes. Mike has argued that we cannot, on the basis of any knowledge about sexual levels of sexual dimorphism, whether it's size dimorphism, weight dimorphism, canine dimorphism, we can't rule out any particular mating pattern or degree of intermale agonism. But I was wondering, does it work the other way around? Are there things that, are there patterns of behavior that forbid certain kinds of dimorphism? And one of the things that we talked about in this connection was the notion of male provisioning of females. Mm-hmm. I was wondering if the two of you would comment very briefly, because we only have a couple of minutes left, on what this implies, what male provisioning of females, as is indicated in Phil's model, would imply about the expected levels of dimorphism that we would see in a population. Phil?
Well, I think there's a strong signal that can be inferred from the lack of dimorphism, or particularly the male feminization of the canine, which really is a strong and unique signal within primates that suggests that there's been this low competition levels. And once we have, say, low competition levels within hominids, there's the potential of there to, once they have a different reproductive or behavioral niche, to actually go to their particular ecological factors infecting size, not necessarily sexual selection.
Mike?
Well, there's two factors there. First of all, the provisioning in modern humans is well accepted as being an important part of the human mating and social system and part of our life history. However, if you look at if Ardipithecus is indicative of being monomorphic in both size and canine size, you have the issue that maybe canine size is no longer being selected with the re-evolution of dimorphism. For example, in Artiodactyls, they've lost their canine dimorphism, and instead of re-evolving it, they evolved antlers and size dimorphism, but you don't have to evolve the two characters in lockstep. So really, you have a conundrum here with, do you use the size dimorphism or the canine dimorphism to indicate behavior?
Thanks, Mike. We'll be talking more about this at greater length, starting over here in the Photonics Center on the Boston University campus at 4 p.m. today, when we continue with the second part of our fifth installment of the BU Dialogues, Does Size Matter? Sexual Dimorphism in Human Evolution. Thanks.